Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Andrew Newland, CEO of Angle, for a conversation on why live cell liquid biopsies should become standard in oncology. Welcome, Andrew.
Hi, Nicole.
So to begin with, I'd like you to introduce yourself a little, Andrew, to our listeners and provide some details of the journey that led you to doing what you do today at Angle.
Yes, of course. Well, I started off as an engineer, so I've got a surf science background, and then I'm also qualified on the financial side. So during my career, I combined those two aspects: the understanding of science and the understanding of business and finance, and specialised in commercialisation of intellectual property. And that led me to the establishment of multiple companies based on very strong intellectual property. And when I founded Angle, the idea was to develop some game-changing technology-based companies which could have a major impact. And during that process, one of the companies that we founded in Angle was actually Parsortix, which is a microfluidic technology that we discovered originating in the United States. But basically, my business, Angle, built the whole commercialization of that. And we found that there was tremendous opportunity to actually recover rare cells from blood. And since we had this microfluidic technology, with a deployment capacity in precision medicine for cancer, I focused all the resources of Angle on that. We sold off our other businesses and reinvested all of it in this microfluidic technology for liquid biopsy.
Thank you, Andrew. Well, that's quite a journey. We're going to break down some of that for listeners today. Let's focus to begin with on live cell liquid biopsies themselves. So for those unaware, a liquid biopsy is a blood test that detects signs of cancerous tumours, including tumour cells and cancer cell DNA. And current US FDA-approved tests can detect some types of advanced cancers, predict prognosis, and help healthcare providers make treatment decisions. Now, these live cell liquid biopsies we're going to be discussing today are a UK-developed technology that aligns with national efforts to modernise cancer diagnostics, as well as potentially reduce healthcare costs by minimising ineffective treatments. So, Andrew, you know my question is going to be, tell us more. Start from the basics and then perhaps get into the specifics of the microfluidic testing.
Yeah, so I think the first thing is why do we need liquid biopsies in the first place? And that is because obviously cancer incidence is increasing greatly right across all different cancer types. And the key thing about cancer is that no two patients have the identical disease. So if you imagine three patients with breast cancer, they will all have cancer in the breast area, that location, but their cancer will be different and it will respond differently to different drugs. So standard of care is a tissue biopsy where some of the cancer is cut out, taken to a laboratory and examined. And from that, the physicians can work out how best to treat that patient. So that's great. And that same approach is applied across all different cancer types. If a tissue biopsy is possible, the cancer is examined and that guides treatment. But the challenge is that the cancer changes over time. So cancer is a heterogeneous disease. It's very dynamic and there's clonal evolution going on all the time. So that the patient's initial cancer will not be the cancer that they have 6 months later or a year later. And consequently, the drugs which are administered may start working quite well for the patient, but at a later point, they no longer are working. And obviously, the drugs haven't changed, but what's changed is that the cancer within the patient has changed. So what doctors would like to do is a repeat biopsy in order to go and look and see what's happening with the cancer right now at this time point. But they can't actually repeat the biopsy because the patient has usually had surgery. So, for example, in the breast cancer example, the patient might have had a lumpectomy or a mastectomy. So that whilst they still have cancer, there isn't a location where there's a concentration of cancer cells that can be cut out and examined. So it's quite shocking, but actually what happens is that oncologists effectively have to fly blind without access to information on what's happened in the patient's cancer recently. So what Angle has pioneered is a liquid biopsy solution to that, which is recovering cancer cells from the patient's blood. So the patient has a simple blood test, just exactly the same as they would routinely have. A tube of blood is allocated to liquid biopsy. We run that tube of blood through the Parsortix system, which is our liquid biopsy solution, and that will recover the cancer cells that are present in that blood sample for analysis. Now, in a standard tube of blood, 10 milliliter tube, there might be 10,000 million blood cells, but only 10 cancer cells. So that's where the technical challenge is. How do you recover a very small number of cancer cells from a much larger number of blood cells? So our Parsortix microfluidic technology has successfully proven that we can do that. And that's based on the fact that the cancer cells are generally significantly larger than the blood cells. And importantly, not just that, but they are less compressible than the blood cells. Blood cells compress to go through very tiny capillaries, whereas cancer cells are not intended to do that. So once you can recover these cancer cells, you can then do all the downstream analysis that you might do with a standard tissue biopsy, but without the need to actually cut anything out. You just do it from a blood test.
Thank you, Andrew. And I believe this is applicable across 24 cancer types. Is that right?
Yes, we have the first and only FDA product clearance for recovering cancer cells for subsequent analysis. And that analysis can be molecular analysis, DNA and RNA. It can be immunofluorescence, protein analysis. It can be enumerating how many cancer cells are there, what type of cancer cells, circulating tumor cell clusters. And our FDA product clearance is specifically in relation to metastatic breast cancer. But for about the last 4 or 5 years, we have provided access to our system to research use customers. And so what I'm talking about here is the world's leading cancer researchers have utilized this system to do investigative work. And there's over 40 independent cancer centers who have published successful results with our Parsortix system. And that covers over 24 different types of cancer. So basically every solid tumor cancer type that has ever been used with, the system works without any modification. So the same instrument, the same one-time use consumable will recover cancer cells for a whole range of different cancers.
And are you able to name any of those cancer centers or their published results at all?
Yes, I mean there's market-leading centres, almost all the ones that anyone might have thought of. But in the UK, for example, Cancer Research UK has done a lot of work with this. Bart's Cancer Institute, University College London, Imperial College, that being the UK groups. In Europe, lots of other places. For example, Hamburg-Eppendorf is a leading German centre, Frankfurt, Dusseldorf, many others, French, pretty much most countries in Europe. And in the United States, many of the leading comprehensive cancer centres, University of Southern California, MD Anderson, Northwest University, and multiple others have used this system. So it's kind of very well established as a research tool. What we're now working to do is to get this adopted for clinical use to treat patients. And that's where further work is needed on the clinical utility of examining the cancer cells.
And can you tell us about some strong real-world evidence from the INFORM clinical trial?
Yes, so INFORM is a trial that Angle has set up and been running for the last couple of years with 6 UK NHS trusts, where they're recruiting patients who have got metastatic disease, breast cancer, prostate cancer, lung cancer, ovarian cancer being the main ones that have been examined. And the aim of that study is to recruit 1,000 patients and to do up to 6 different time points, because one of the critical things with a blood-based liquid biopsy is that you can monitor patients over time and look at the changes. And indeed, we've already demonstrated that there are very significant changes that can be seen by analysing the cancer cells that come out at those different time points. For example, in breast cancer, one of the most important targets is a protein called HER2. So this is something that you can measure on the cancer cells at standard diagnosis, the standard tissue biopsy. One thing they will look for is HER2 positivity or not. And if the patient is HER2 positive, then they will be prescribed with a HER2 based targeted therapy. Trastuzumab is, otherwise known as Herceptin, is a very well-known first-line therapy for HER2 positive breast cancer. So that's a standard of care. More recently, AstraZeneca's drug Enhertu has been cleared for use with HER2 positive and HER2 low breast cancers at later stage. So this is second or third line therapy. So interestingly, the clearance on Enhertu, which is a big drug, I mean, something like $5 billion a year revenue line for AstraZeneca, is limited to patients who are HER2 positive or HER2 low in their tissue biopsy. But of course, the tissue biopsy may have been taken 4 or 5 years before the prescription of the Enhertu drug. So one of the things that we've seen with our INFORM study is measuring the HER2 status on circulating tumor cells, so the cancer cells from blood, for patients who are HER2 negative by tissue biopsy. So these are patients who, because they're negative by tissue biopsy, would never be prescribed Enhertu under their label. But we've discovered that up to 40% of those patients can be HER2 positive in their circulating tumor cells at a later time point. That's very significant. And unfortunately, we've already seen 4 patients that have come through our testing, which is obviously for research purposes, found them to be HER2 positive. They were never given a HER2 targeted therapy, although you could conclude they might have benefited from that, and they subsequently died. So our mission is to get this new approach of examining the cancer cells to look and see what the current status is to guide treatment going forward. And we've done a lot of extra work, not just in proteins like HER2, but also in DNA mutational status, where we've got DNA sequencing assays in place now, which can measure, well, literally thousands of different DNA variants, but there's probably a dozen or more with targeted FDA-cleared therapies associated with them. So we can measure that on the CTCs. And what we want to do is clinical studies to get that adopted as standard for patients. So patients should have, and not just breast, this is right across all the patient types, they should have the benefit of blood tests to guide their treatment.
I wholly agree. So my final question actually is, if we're looking to the future of cancer care and everything we've been discussing in this conversation today with these liquid biopsies, what does the oncological horizon look like, Andrew?
What I see in the future is that there certainly will be standard routine monitoring of patients, probably every 3 months via blood tests, to examine, firstly, are they responding or not to the drugs that they've been given? And that can be measured by looking at the number of circulating tumor cells and their composition to see if that's changing. And secondly, is the target for the drug still present in the living CTCs or not? If it is, continue with the drug. If not, then look for other targets. And by doing that, I believe that patients will have better outcomes. They will benefit from being given drugs more likely to help them. And remember, all the drugs that patients receive have negative side effects with them, toxicities, etc. So it's not only not helping them to be given a drug that's not working, but it's actually hurting them. So we want them to have drugs that will benefit them. But there's a massive benefit for the healthcare providers because the cost of these drugs is extremely expensive. So new drugs can cost £250,000 per patient and yet only work for 1 in 5 patients. So by adopting this personalized cancer treatment approach, it can also reduce the overall expenditure of healthcare resources on cancer care and make sure that it's better targeted.
Thank you, Andrew. It's been a pleasure.
Thank you very much, Nicole.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.